So when the first genomes of eukaryotes have been sequenced, the surprise was very big that most of the DNA does not encode for proteins, but it's actually what we call now non-coding. And from this non-coding fraction, a large portion corresponds to mobile genetic elements or remnants thereof. And small RNAs are very important in controlling the relocation, or how to say, the spread within the genome, and they basically limit the transposable elements to where they are right now. And this is in particular very important in the germline, where reproductive cells are formed, because errors that happen in the germline will be passed on to the next generation, and this can be very detrimental. And in our group, we are interested to first understand how small RNAs are actually made, how are they produced within a cell, and secondly, how do they elicit their function. So the biogenesis of small RNAs involves more than 30 factors. And so far we only understand a few single factors, what their role and what their function is. But we really want to understand what's the interplay between the individual factors, so how do these factors come together to elicit their function, and how are they regulated. So enzymes need to be tightly controlled, so they should only be active at a certain location in the cell at a certain time point. And only if you really understand very precisely how an enzyme or how proteins work, you can also make use and manipulate them even more precisely to ask other questions that you couldn't ask before. I'm a biochemist and structural biologist by training, and we want to understand how proteins or complex of proteins accomplish very complex tasks within the cell. And to understand this, very often you can understand problems if you look at them very, very detailedly and with large magnification. And in structural biology, we try to determine structures of proteins or architectures of protein assemblies in order to figure out what they're actually doing. So the fascinating thing is that nature usually have come up with several solutions to one and the same problem. And for example, genome defense systems are already present in bacteria, as they are in humans, and they are conceptually very similar, but the mechanistic detail differ quite tremendously. And the interesting thing is, if you understand the system from one organism, to try to understand the system in the different organism and then to compare it. So science is never ending. So answers that we find will generate more and more questions, and thereby you never stop learning, you learn new methods, and you always find new questions to answer. So at one point you find a question that you find interesting and that you want to understand, and then it takes a long time to collect all the data and information that you need to answer this question. And the most motivating moment, I guess, is when you have all the data together, all the puzzle pieces at the end fit together, make sense, and you can answer questions. This is really motivating to me. But the same motivation, I would say, comes from training of students. So to train students to become independent researchers, that they are able to make experiments and address questions without actually needing you. And if they then come up with results that they generated independently, this is also something that is very motivating. Just follow up what you're really interested in. Try to find out what interests you, what motivates you, and what comes easy to you. So if you find, so I knew that science, chemistry, biology came very easy to me. So I didn't need to study much, and it was very easy for me to understand, like quite intuitively. And if things come easy to you, the chance that you will be successful are very high. So especially also if you enjoy what you're doing. So just try to find out what you really want to do.